preview.
As what we have all known, all living things are made from cells. Each of these cells may be a plant, an animal, a fungus, or bacteria, all have a cell membrane.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example.
Cell membrane is a semi-permeable membrane and have control over what molecule can or cannot pass through. It serves three important functions for a cell. First, it protects the cell. Second, it regulates the transportation of molecules and ions. And lastly, it communicates with other cells. A cell membrane is made up of components and different structures. These are the phospholipids, proteins, cholesterol, and carbohydrates. The phospholipids consist of two parts, the hydrophilic, which is the head that likes water, and the hydrophobic, which is the tail that doesn't like water. Lipids are packed tightly together. Only tiny molecules or ions may move without any problems between the inside and outside of the cell. These molecules or ions must be slightly hydrophobic or water repellent to move through the cell membrane. Proteins, which are also known as macromolecules, are composed of amino acids. They comprise a large part of a cell membrane's components and regulate the movement of molecules and ions, including the nutrients in and out of a cell. Surface proteins float laterally like boats on water and may serve as markers that enable cells to recognize each other or as receptors of signals from other cells or molecules. When you hear the word cholesterol, the first thing you can probably think is that it is bad for the health. However, cholesterol is actually a very important component of a cell membrane. Cholesterol molecules are made up of four rings of hydrogen and carbon atoms. They are hydrophobic and are found among the hydrophobic tails in the lipid bilayer. Passive transport is a kind of transport that doesn't require energy, and it has three kinds as an inactive energy transport. These are diffusion and osmosis. Diffusion is the movement of particles down to their gradient on the molecules from a higher concentration to a lower concentration.
Passive transport means it's an automatic process that doesn't require any input of energy. For example, diffusion is a passive process in which particles move either into or out of the cell from an area of higher concentration to an area of lower concentration. The cell doesn't use any energy when this happens.
Simple diffusion is the unassisted passage of small, hydrophobic, nonpolar molecules from higher concentration to a lower concentration. Very small molecules can slip through the cell membrane too, even if they are hydrophilic, just like a few ants might crawl through a crack in the wall just because they're tiny. So how do large, charged, or hydrophilic molecules pass through the cell membrane if they can't simply just diffuse in? Well, think of how your friends come into your house. Under most circumstances, they'll use a door. A package delivered at your residence would come through your door too, but it would need someone to carry it in. A fly might come through an open window on its own, while a squirrel could come down your chimney. There are different types of passageways into the cell, just like there are different ways to get into your home, depending on who or what is trying to get through. So each method of passage through the cell membrane might be useful to different molecules. So, facilitated diffusion is passive transport that uses integral membrane proteins to help larger, charged, hydrophilic.
Facilitated diffusion is the process of transporting particles into and out of a cell membrane. It is the movement of substances from a higher concentration to lower concentration with assistance. This assistance comes from proteins embedded in the cell membrane. They act as a channel or a carrier, allowing the passage of needed substances that can't diffuse on their own. Osmosis is the movement of water which can easily pass through the cell membrane. It changes the cell volume by depending on the direction where water flows across a cell membrane. Osmosis can cause a cell to shrink or swell. There are two types of solutions where the volume of an osmosis changes. These are hypotonic and hypertonic. And what do these two differs all about? Transport is a kind of transport that requires energy, or also known as the ATP, which stands for adenosine triphosphate. Active transport has three kinds. These are the sodium-potassium pump, endocytosis, and exocytosis. Now, let us take a look of this video on how an active transport works inside a cell membrane.
The second category of cell transport is active transport. This is when particles move from an area of lower concentration to an area of higher concentration. When particles move against the concentration gradient, energy is required, often to allow protein pumps to assist in particle movement. Why would the cell need to move particles from a low to high concentration and expend energy to do it? An important example is seen in your heart muscle cells. In order for your heart to beat, there are certain molecules that have to move from an area of low concentration to an area of high concentration for those cardiac muscle cells to work.
Membrane pumps, or known as the sodium-potassium pump, which is a carrier protein from a low concentration to a high concentration.
The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Endocytosis is a process which cells ingest macromolecules. It comes from the Greek word endon, which means within, kytos, which means cell, and osis, which means process. This is the process where a substance is brought inside the cell that have to pass through a cell membrane. Let me show you how an endocytosis gets done inside the cell membrane.
During endocytosis, the cell membrane folds into a pouch surrounding particles outside the cell. A vesicle is formed. This vesicle carries the particles into the cell and can release its contents into the cytoplasm.
Endocytosis is divided into 3 parts. These are phagocytosis, pinocytosis, and receptor-mediated. Phagocytosis, from the word phago, comes from the Greek word phagin, which means to eat. This is the process which cells engulf and digest large molecules. The pseudopods surround the food item and fuse together, forming a residue to take the food into the cell. Pinocytosis still comes from the Greek word pino, which means to drink, where a cell takes in fluids. The vesicles of fluid are formed in the cell membrane without the formation of pseudopods. Receptor-mediated is a process that recognizes and targets a specific substrate through the help of integral membrane proteins on the cell membrane surface. Receptor-mediated have special protein peaks in the cell membrane and are lined with receptor proteins that bind with specific materials. Exocytosis. This is a process by which a substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. Here is how an exocytosis works.
Exocytosis is the opposite of endocytosis. Particles are transported out of the cell. A vesicle containing wastes or cell products moves toward the cell membrane. The membrane surrounding the vesicle fuses with the cell membrane. The contents of the vesicle are secreted.
Thank you for watching, and I hope you learned something out from this educational video lesson. Good day ahead, everyone!